The first step of gene expression is transcription. This is when a gene is copied to form RNA with the help of RNA polymerase. There are three stages: initiation, elongation, and termination. Prokaryotic cells need an initiation factor in order to begin. A single RNA polymerase is responsible for transcription in prokaryotic cells. To initiate transcription at a promoter site, polymerase needs an initiation factor sigma, which is a subunit of RNA pol that is required for binding to the promoter region. Initiation. Sigma and RNA pol bind to the promoter and form a closed promoter complex.
Like so.
The tight binding results in local melting of the double helix, and the closed complex is converted to an open promoter complex.
Like so.
RNA polymerase incorporates the first few BP, but polymerase remains at the promoter. Promoter clearance, the sigma factor dissociates from the complex and elongation phase begins.
Peace out.
Elongation. RNA pol adds one nucleotide at a time and has two proof readings.
Pew. 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 Pew.
Termination. Role dependent. A ring-shaped protein binds to mRNA and follows along RNA pol. When hairpin forms, DNA pauses, Rho acts as a helicase and causes the DNA and RNA to unwind and dissociate, releasing the mRNA. Translation is the synthesis of proteins. There are also three stages to this: initiation, elongation, and termination. Initiation factors include IF1, which prevents tRNA from binding to site A, IF2, which is GTPase, and IF3, which temporarily blocks large subunits. We also need ribosome binding site, with an A site, and a P site, and tRNA, which is located at the P site, and with its base pairs of the AUG codon. Elongation. Initially, the amino acid tRNA is bound to the P site on the ribosome. Elongation begins when a second AA tRNA binds to the A site on the ribosome, which requires the use of EF2, which is an elongation factor, and GTP for energy. This helps escort the tRNA to the binding site.
This way.
Peptide bond formed between the first AA and the amino acid part of AA tRNA in the site A. This is done by the enzyme peptidyltransferase. Result: dipeptidyl tRNA in site A. Transfer RNA with its amino acid will move toward A site. If the anticodon matches the codon of the mRNA strand, amino acid will be transferred to the P site. The transfer RNA will exit out of the E site. Then it will move along the mRNA strand and continue its pattern.
Termination of translation. There are three stop codons: UAG, UAA, and UGA. Their role is to terminate the polypeptide sequence. Once this occurs, the ribosome dissociates, and stop codon and polypeptide sequence are released.